Today, my title is Microfluidic and Spleen-on-a-Chip Studies of Sickle Cell Disease. So first, some disclosure. I have some financial relationships with Global Blood Therapeutics, GBT, which is part of the Pfizer now and Analytica. Red blood cell is a simple model cell without nucleus. So these red cells, the basic function is that they deliver oxygen from the lungs to the body tissue and take CO2 back from the body tissue to the lungs. So it undergoes severe reversible large elastic deformation, especially through the smallest capillary and the spleen slits. These are really small openings, and they have to deform. It's a 8 diameter disc has to deform through these very small openings on the order of 2 to 3 micron. So it has to go through large reversible elastic deformation. And that's why compromise of red cells deformability can cause many diseases. So that includes sickle cell disease. The lifespan of normal RBCs is about 120 days. So in that 120 days, it takes about 500,000 circulations, which is about 1 minute per cycle through the whole body. Sickle cells would have a much shorter lifespan. It has 10 to 20 days lifespan. Still, in that short lifespan, it still go through 40 to 80K circulations. So there's a lot of fatigue cycles mechanically, and also a lot of cycles of hypoxia, normoxia cycles. So these repeated challenges could bring some damage to the red cell. So we'll go through these in detail. Sickle cell disease lead to acute and chronic complications. It's a single amino acid disease, which is well studied so far. However, there's still a lot of things unknown, including it's hard to predict vaso-occlusive crisis or acute splenic sequestration crisis or ASSC. These crises are caused primarily due to the cell sickling. The sickling is caused by sickle hemoglobin. They polymerize and grow stiff fibers under hypoxia. So this is the root cause for sickle cell disease. That's why we need to study under hypoxia conditions. There's a lot of things still unknown, especially when the vaso-occlusive crisis would happen. So we have developed a microfluidic platform for studying red cell or sickle cells. So we initially the dielectrophoresis stretch assay to stretch the cell to test the deformability in the microfluidic channel. And then eventually we can do this multiple times, and this is testing fatigue. This setup we have actually can do transient hypoxia. So you can do hypoxia, normoxia within a relatively short time. So you can do this back and forth for many cycles. So eventually we combine everything together so we can do repeated hypoxia and in situ mechanical loads. And we can also test mechanical properties in situ. So we developed a single cell microfluidic based experimental platform that can induce single or repeated transient hypoxia cycles, can impose in situ mechanical, including fatigue loads, and also test the mechanical properties of red cells in situ. Okay, so this is a slide showing some results about mechanical fatigue. Under the same loading, you can see this cell initially stretched to this much. But when you go through about 900 cycles, under the same electric field, which is the same stretching force, it actually stretched to a shorter length, which means under the same force, it can only stretch to a shorter length. That means it's getting stiffer. So after repeated cycles, the cells become stiffer and stiffer. So mechanical fatigue can make the cells lose its deformability. So these are some results showing the stretch relaxation curve. So under the stretch, it will stretch to a certain level under different forces. You can see after 900 cycles, the maximum stretch will decrease. And this is the relaxation part when you release the load. And then this part, you can actually extract the viscosity of the cell. So we tested that the stiffness will go higher and higher with number of cycles. And depending on how high the load is, the higher the load, the faster the degradation. Similar trends can be found for the increased viscosity of the red cell. So we found similar deformability degradation versus fatigue loads for sickle cells as well. So this is a slide showing repeated cycling kinetics and cell shape evolution under repeated deoxygenation oxygenation cycles. So you can focus on this cell, for example, between two different cycles. You can see each time when it deoxygenates its sickle, and when you reoxygenate, it will unsickle. But in between two cycles, this same cell will sickle into different shapes. So it's a stochastic sickling process. Between two different cycles, it will sickle into different randomly, kind of random shape. So this is the oxygen level changes between these two cycles. And you can see that we can actually account for the using deep learning to quantify the sickle fraction versus time. So this is the sickling kinetics curve, because there's a lot of heterogeneity. You can see that even these cells are from the same sickle cell patient, but they don't behave all the same. Some sickle faster, some sickle slower. So that's why we have the sickle fraction versus time curve. You can quantify this. And we did find that actually the sickle fraction will go up after each cycle. So there's a degradation over time. The same thing as mechanical fatigue. This hypoxia fatigue also, accumulate damage for the sickle cells. And the delay time also goes down. That means it sickles faster, a little bit faster each time. So we actually studied the effect of Voxlotor treatment under cyclic hypoxia. So this is with and without drug treatment. This is a schematic showing that with drug treatment, you will see the sickle fraction will go down versus time. So this is the results, sickle fraction versus number of cycles. 30% modification is the drug dosage achievable in vivo in the human body. And this is actually an FDA-approved drug. And without treatment, you can see the sickle fraction goes pretty quick after each cycles. But when you have anti-sickling drug treatment, this is much more suppressed. That means the degradation is much slower. So this is at T at 20 seconds, and this is showing at 60 seconds. It's at 60 seconds, you also see a big difference. When we have repeated hypoxia cycles, what happens with the mechanical property degradation? This slide shows that shear modulus versus number of cycles. You can see that even healthy cells will degrade a little bit over these hypoxia cycles, but sickle cells actually go much worse. And these one interesting observations is that because we have a single cell assay, the cells, you can divide them into the cells that actually sickled and the cells not sickled. These are from sickle cell patients. You can see that the sickled cells really degrade very fast, but those that actually not sickled, they don't degrade that fast. So this is why this is important to use anti-sickling drug to control the sickling. Similar trends happens to the degradation for the cell viscosity. The cell viscosity increases much faster if you count for the cells that actually sickled versus those are not sickled. So what happens if we add anti-sickling agent 5-HMF? This is the shear modulus versus number of cycles. This figure we just showed that without anti-sickling drug. But if you treat it with this 5-HMF as an anti-sickling drug, you can see that with reduced sickling, you can mitigate the degradation of these sickle cells. Similarly, we can see the protection against the viscosity. The viscosity changes very fast without drug here. With drug treatment, you significantly slow down the worsening trend. Okay, so next we'll discuss a little bit about the spleen red cell retention and elimination. So in the spleen open circulation, when the cells go into this open circulation, then these red cells will see a lot of these residential macrophages. They can be adhered and eaten those altered red cells. These old cells can be taken out by these macrophages. On the other hand, the spleen slits or interendothelial slits, IES, these small slits can mechanically challenge these red cells. If they're stiff, then they cannot go past. They will be retained and blocked. And these blocked cells will also eventually eaten up by these macrophages. So in the sense, we need to actually study the balance, how fast these retained red cells can be cleared. If these cells, these incoming cells continue to come in, if those retained cells that cannot go through the slits and back into circulation, and if they're retained, they cannot be taken out, eliminated by macrophages fast enough, then there's going to be some sort of congestion. So the balance between RBC retention and elimination would be very important. So on the other hand, for sickle cell disease, there are studies that identify that in the spleen, there are sickled cells. That means cells being sickled under hypoxia. So the spleen has to be somewhat hypoxic. So we need to study the hypoxia condition in the spleen. So to do that, we did Spleen-on-a-Chip studies, and we have designed this S-chip that cells going through these small slits mimicking the IES. or interstitial slits. And on the other hand, we also study the macrophages under slow flow. These macrophages can return and eat some of these sickle cells. So we will study this for sickle cell disease. So this is the under normoxia for healthy cells. You can see these slits don't really retain any red cell. On the other hand, if we have sickle cells under normoxia, you can see there are some accumulation here, quite a bit more retention even under normoxia. But if we have sickle cells under hypoxia, we can see what happens. It can quite quickly get clogged because this sickle cell getting sickled and getting very stiff, and then you can quickly block all these slits. So these observations, we kind of observed all of them either in vivo or in vitro or ex vivo by our collaborator, Dr. Pierre Buffet. He has the setup to do these tests and to do histology observations of these different conditions that mimic what's observed. So we can see that the number of open slits versus time for different conditions for healthy cells, AA, oxy, AA, deoxy, these are healthy cells, heated cells, sickle cells under normoxia, sickle cells under hypoxia. So under hypoxia, the sickle cells really being blocked very quickly. That means the number of open slits drop down very, very quickly. On the other hand, these sickle cell under normoxia, they also drop down quicker than the healthy cells, but they're much slower than these under hypoxia. So, for example, if we pick 40 seconds at the time point in our microfluidic device, you can see the number of open slits under different conditions. The AA oxy versus AA deoxy, that means healthy cells under oxy or deoxy condition, they don't statistically differ that much. However, compared to sickle cell under oxygenated condition, there's a difference. There's a much bigger difference if you have sickle cells under hypoxia. Actually, the number of slits open are almost zero at 40 seconds. So this is the difference under different conditions. So one interesting observation is after the blocking of all these slits, if we put under hypoxia, if we put back the oxygen back, so it became normoxia condition, what happens? This is what happens. When you put oxygen back, it will unsickle these sickled cells and unblock a lot of these blocked slits, and it can start it to flow again. So this somehow mimics what's happening in actual medical practice. These doctors would put transfusion, or when you have a severe splenic sequestration crisis, transfusion, these transfused blood will bring oxygen to the spleen and can relieve the emergency situation. And that means when you bring the oxygen to the organ, that will help unclog the spleen. On the other side, what happens to macrophages? So adhesion of sickle cell to macrophages under normoxia and hypoxia, these are interesting questions. So this is the video going to show under normoxia, the retention and adhesion versus sickle cell. But these are macrophages. And this is the under hypoxia, the same patient sample. You can see that after the same amount of time, that there are a lot more adhesion and retention on these under hypoxia condition to retain sickle cells. So this is the summary of the number of the attached RBCs to macrophage. So healthy cells really don't adhere that much, but sickle cell under oxygenated condition, there will be a little bit of adhesion, but under deoxygenated conditions, there will be a lot more adhesion. Our collaborator Guangxin Li and George Kalandakis, they actually studied digital twin of this. There's a lot of simulations we can do to mimic this. So after retention of these sickle cells under slow flow, mimicking what's happening inside spleen. So sickle cells under normoxia, it will getting eaten. You know, there's a process of being pinched and then eaten. So this is the at about 12 minutes. This one shows it's being taken inside. For sickled red cell under hypoxia, it's interesting that after taking inside, it's actually still sickled. But the interesting observation is the complete internalization takes about only 7 minutes. So the internalization goes pretty fast under hypoxia. So if you quantify this, we can see that internalization time for non-sickled normoxia versus non-sickled hypoxia. So it's interesting that only for those sickled under hypoxia, there's some cells, sickle cells, they don't sickle under hypoxia. Then they're eaten, the internalization speed does not really change that much versus the under normoxia. But on the other hand, the sickled ones that actually sickled these will be internalized pretty quick. Again, we can also do digital twin for these internalization process. So this is an actual video showing that the sickle cell being taken by this macrophage. You can see this sickled cell being internalized. The interesting thing is it's being internalized and it will stay sickled. And this sickled cell can stay sickled inside for a long time. It's being hard to being digested. It takes a very long time to be digested. So if you observe these different conditions under microscope, this confocal microscope, you can show sickle cells. This is under oxygenated condition, and under deoxygenated condition, when they don't sickle, they behave quite the same. But under deoxygenated condition, they stay sickled for a long time. They sickled and being taken sickled on the hypoxia. It's very interesting. So these will stay long time not being digested. It's very difficult to digest these polymerized fiber in this sickled cell. So this takes long time, and this about half of this happening in this situation. So the next one we are going to show is that the ingested sickled RBCs after reoxygenation, what happens? So this is a sickled cell being ingested, but after we put oxygen back on, it will unsickle. You can see it was unsickled, and then it was actually broken up pretty normally as the under normoxia. So if you put oxygen back on, this can, again, to help you speed up the digestion process. That's a very interesting observation. So in summary, I think we studied different scenarios and different situations of the spleen retention by spleen slits and by spleen macrophages. So under healthy conditions, the retention by the slits or macrophages versus the elimination speed, they can be balanced because there's a very little bad cells being retained. And there's these little small number of bad cells can be taken out quickly. So they will be maintained balance. The spleen slits remain open and flow through the red cells, no problem. Under stable sickle cell disease, we can see that there are more sickle cells will be retained faster. But also, these sickle cells can be taken out quicker, adhered quicker, and eaten quicker. As long as they remain balanced, then it may cause anemia because it may overwhelm the red cell generation by bone marrow. So that may cause anemia, but they may not cause any spleen swelling. So under hypersplenism, what happens is the retention of the sickle cells can be higher than the capacity of eliminating these cells. The spleen works very hard, but the macrophages cannot keep up. So then the balance is being broken. And in this case, the anemia will be more severe than this situation. And also the accumulation of red cells in the spleen, because you cannot keep up with elimination, there's always more red cells being accumulated in the spleen. Then it will cause spleen swelling. So this is what happens. But when this situation persists, and this could trigger a vicious cycle, then a sustained accumulation of red cells in the spleen can cause worsening hypoxia conditions, and then more sickle cells getting sickled and become very stiff and more adherent. And but on the other hand, you have a slower, when these are taken up by the macrophages, the macrophage can be very slow to digest this sickled red cells despite faster ingestion. So this will cause more severe congestion. So this becomes a vicious cycle, and so in that case, this retention rate is much higher than whatever the elimination rate can keep up. So will cause severe anemia and severe splenomegaly, and it will be a life-threatening crisis. So this, you can see more details in this PNAS paper we published recently. So in summary, we established a single-cell microfluidic assay that can induce single or repeated transient hypoxia cycles in post in situ mechanical, including fatigue loads, and test the mechanical properties in situ. And repeated hypoxia cycles can induce accumulative red cell deformability degradation and sickle in both healthy and sickled RBCs. The effect is much stronger for RBCs that sickle under hypoxia. So anti-sickling drugs can mitigate the repeated hypoxia-induced mechanical degradation and cell damage. Spleen-on-a-Chip, we studied this and found out that retention versus elimination of sickle or red cells, this needs to be balanced. It's important to keep a homeostatic balance. So what the platform can do is can be used as drug efficacy and diagnostic assay for precision medicine. I would acknowledge my main collaborators from MIT and other institutions and key contributors over years in my lab, and the funding support from NHLBI and GBT. So I'll stop here. Thank you very much.